Okay, I think we're recording. The sound is good this time. So today, we're going to talk a lot more about the visual pathway. This is a continuation of what we talked about last time. Last time, we started talking about sensation and perception. And kind of, you got to start from the physical world, talk about what are those senses that we have evolved as humans, and what can we actually sense in the world. And then based on that senses, we could build up our perception. Um, Vision being one of the most important ones, and we kind of consider ourselves visual animals. Um, so we'll spend a lot of time on the vision. And as a case study too, right? It's stuff that you learn about visual pathways, somewhat generalized to other modalities in some ways. But the principles are there. So we're going to spend a lot of time on visual pathways for a few weeks. And build our way up to object recognition. There are two main pathways. We're going to spend some time on one, which is the pathway that takes visual input from the eye and then takes it all the way to the visual cortex. There's another pathway that takes input from the eye and takes it to the superior colliculus, and it's very instrumental in eye movement, and we're going to talk about that later. So last time we talked about vision being really about these sorts of visible light. And we talked about what makes certain kind of electromagnetic signal be visible, some other not be invisible. And that is basically the wavelength where our eye has evolved to detect. Right? So again, this is the first step. You have to have a sensory input that could detect something. If you wanted to have a if you wanted to have a light that, like, if you wanted to put a lamp outside your house that lights up when someone walks by, right? What is the first step? What would be the first step for you to think about? If you were faced with that problem? What's that? Sensation, right? If I said you want to put a lamp that lights up when somebody walks up, that means that whatever system you're building has to sense something in the world, right? If that thing, is supposed to work, it has to sense something. That's the first step. In the example that I gave, what would be that sensory device that you would use? Yeah. Movement detector. Based on what? Changes in light that bounce off of But let's say it's a dark environment already. Yeah, the light isn't on yet, right? So in a dark environment, like in New York, you walk by a house and the light comes on. What kind of sensory input are they using? Like a beam of light that you break that beam of light. Okay. Like a non-visible beam of light that you break, and that triggers the. Could be that, yeah. Or something we talked about last week that every human kind of generates. What is that? Heat. And it's what kind of light? Infrared. So those are infrared lights, right? So you walk by, it detects maybe some infrared light coming, and if it detects an infrared coming, it could turn on the light. It's a very simple system. Infrared cameras, right? They basically detect all the heat in the environment that comes from bodies, and it could light up. So why am I giving you that example? Because you have to always think about the first step is sensation. You're sensing something in the environment, and then based on that sensation, you could act. The device that senses a human walking based on infrared signal and lights up, turns on a lamp, is a good. It's a device. It's like a full device. It senses, it recognizes something, and it also acts on it. So it's like the entire spectrum. So that's, yeah. Think about it that way. Now, the function of the eye is to focus this energy in the visible spectrum and transduce it to neural signal, and then start processing the signal. That's what the eye does. And the eye is so important for many reasons. And there is a quote here by Da Vinci, the eye, which is called the window of the soul, and you hear that metaphor a lot, is the principal means by which the central sense can most completely and abundantly appreciate the infinite works of nature. So if eye was always fascinating, right, to people, how it does, how it functions. But a lot of our actions in the world, right, your eye movements, right, we make eye movements 3-4 times a second. A lot of our head movements, body movements, all of that is really to bring something to your eye. That's what you're doing. So we're really really a lot of our actions and perceptions revolves around it. But you already know that. So when you look at the eye from the outside, it looks like this. You don't need to know the areas. Just know that the light travels through the pupil. That's all that matters. The other areas are not really letting the light pass. When you look at the structure of the eye, and it's a very fascinating structure, and we're going to unpack it because there's a lot going on here. But so if this is the eye, and you see there is a lens, right? Like a lens that you have in a camera, there's a lens in the eye, which is right in the pupil. That's where the area that the light can actually pass. Then this is the body of the eye, and the light goes through here and reflects in the back of the eye. Now, if we take this very tiny triangle, rectangle that you see here, and blow it up, you get a figure like this. If you blow that up, you could see that the photoreceptors are really at the bottom of that. Again, this is the most peculiar thing about the eye, that the photoreceptors are on the inner layer of this membrane tissue of the retina. And the light, as you could see here, has to travel through ganglion cells and through middle layer cells to reach the photoreceptors. Now, you would think a better system would be what? Well, the light comes, so when the light comes through the eye, the photoreceptors would be out here. They would connect to middle layers, they would connect to the ganglion cells, and then they would leave the eye, right? That would be a better system. But the design is inverted, meaning that you have to go through the ganglion cells, through the middle layer cells or bipolar cells that we talked about, and then reach the photoreceptors, right? That's a very peculiar thing about the eye. What is the main problem that it causes? The main problem that it causes is that what is supposed to happen to all these axons leaving the ganglion cells? So if you have, let's say the light comes through, goes through all these cells to reach the photoreceptors, the ganglion cells are out here. All of these axons have to leave the eye somehow. So that happens here. So imagine these yellow stuff are the ganglion cells. Does that make sense to everyone so far? What the problem is? That these are the ganglion cells, and the ganglion cells have to just bundle together and leave the eye through the optic nerve. What problem does that cause? Well, it creates a hole in the retina. Does that make sense to everyone? So if I have to bundle all these cells and leave, in this area, you cannot have any photoreceptors. The photoreceptors are down here. Does that everyone understanding this? Photoreceptors are on the inner layer. The light travels, reaches them, but all these nerves, all these axons from the ganglion cells have to bundle together and leave the eye. So there is an area in the retina that there is no photoreceptors. And if there is no photoreceptors, as I said, if there is no receptors, there is no perception really. And we're gonna do a demo. Okay, so I want you all to put up your left thumb okay? And close your left eye. Then move your right thumb toward your left thumb. Keep your eye on the left thumb. There must be a place where the right thumb disappears. Do you get that? Very close. No, you don't have to go that far. Left eye is closed, right? The right eye is looking at the left thumb. And then the right thumb is moving toward it. Yes? Does anyone get it? No one got it? Oh my God, this, all right, we'll do it again. Do it with the index finger. Do it with the index finger. So close your left eye. Close your left eye. It has to be closed. With your right eye, look at the tip of your left index finger. Now move your right index finger toward it. Go until you're about like 5 inches away. While you're looking at the left index finger, in your periphery, the right index finger should disappear. The tip of it. Yes? Yeah? Okay. Anyone not get it? You could say it if you didn't get it. But yeah, if you know where it is, it's very easy to find it. But one eye has to be closed because where the blind spot falls for the two eyes are in different locations. So if your two eyes are open, one of them fills in the other one. So it should be something like this. This is where the blind spot is this is the blind spot for my right eye, where this finger is. Yeah? I think you got it. So where that is, that's where the ganglion cells, the axons from the ganglion cells are leaving the eye. So the eye has a hole in it. And the problem the reason the hole exists in the eye is because of this organization. I want you to understand the relationship between anatomy and function. So because you have this anatomy, because the photoreceptors are at the bottom and the light has to travel, and there are cells above of them, and there are cells above of them, and all these axons have to leave the eye. So at some point, they have to leave the eye. And at that point, there is no photoreceptors. A hole in the retina through which the ganglion cells axon leave the eye and travel to the brain. That's what a blind spot is. We are blind at this location in our visual field due to the absence of photoreceptors at the eye. Okay. Now, you can't do it really because you have different distances from the screen and everything. You can't do it. It's much better to do it in a distance like this as you did it. But I have these slides so you know what it is. Now, you don't see, what's important is that even when you have one eye closed, and if you don't do this experiment, you don't know there is a perceptual hole there, right? Because even when you're looking with one eye, perceptually you're filling in stuff. It's like you're not aware of that there is an area in the visual world that you're not getting the visual input from it. So instead of seeing a hole in your, in your visual world when you close one eye, instead you just fill it in with the surrounding area, and that's what we call perceptual filling. So another thing about this anatomy that is interesting is that the distribution of photoreceptors change across the retina. So when you get closer to the fovea, and we'll talk about what fovea is where the central vision is. When you get closer to the fovea, you're going to have a lot more cone receptors. And when you're farther from the fovea, and then we call that what periphery, right? You have central vision, foveal vision, and they mean the same thing. And then you have peripheral vision, and there are other names for it, I'm sure. But peripheral vision, you have more rods. So the type of receptor that you have in the fovea and outside are different. And these are the schematics, and just if you're curious, These are how they're usually displayed. A cone receptor, which is more packed near the fovea versus the rod. And under a microscope, they kind of have that shape. So it's inspired by the way they look like under a microscope. This is the distribution. So if the angle 0, if the point that you're looking at, that's always the point. That's the foveal point. So if I'm looking at the tip of my index finger, the visual information from here reflects on my fovea. That's what it means, right? That's your fixation point. That area of the retina is packed with cones. So this is a distribution. The area near the fovea is packed with cones, and the area in the periphery is packed with rods. Now there are little cones in the area far from the fovea as well, but they're not as many. Most of them are in the fovea. And then the rods are outside. Now, something that is very important about cones and rods is that they have a very different convergence factors. Now, this is something we talked about. Remember the context of it. If I said if a neuron has a large convergence factor, it means what? What does it mean for a neuron when it has a large convergence factor? Bigger receptive fields, because it's getting input from a lot of different other cells or receptors or something like that. Now, if you have a smaller convergence, it means that you're not averaging too many things. So the relationship between convergence and spatial resolution is what? If you have a higher convergence, is your spatial resolution good or bad? Good. Why? What is a spatial resolution? When we talk about resolution, what are we talking about? Okay, yeah, you're on the right track. So when you're averaging, if I am in this room and 5 people are talking, right? I'm averaging all the input from all of them. This is a high convergence, which means that I'm not going to really understand individuals, what they're saying. So high convergence translate into losing high spatial information. Because I'm looking at a lot of people, I'm listening to a lot of people, it means that I am not able to retain high resolution information coming from each of them. But if I'm only listening to one person, then I would be able to understand the details of what they're saying. So this is the relationship between convergence and resolution. So if, for example, so if I say the cones have low resolution, it means that, or low convergence factor to ganglion cells, it means that what? It means that not that many cones are talking to one ganglion cell. If I say rods have a high convergence factors to ganglion cells, it means that a lot of rods are talking to one ganglion cell. The result is that you would have lower resolution of information in the periphery and higher resolution in the fovea. That's why, like, when we did an experiment and I said, well, look at here, for example, just keep your eye on most and try to read this sentence, right? When you look at most and try to read the sentence, you can't really go that far, because in the periphery, you're really averaging a lot of information from a large areas, and you lose those high resolution information you need to read, because letters are very similar to one another. Like an E and an A, if it's in the periphery, you cannot tell which one it is. But if you're looking at it, you could tell which one it is. So it's at a high resolution when you're looking at it. Does that make sense to everyone? Now, this is a schematic. let's say you had this visual input. There are two lines. If you're averaging all these responses with high convergence, you get an output. If this line was connected to this one, you would get the same input because we talked about it. Let's say these are all excitatory. If this line here was connected to this one, you would still have the same amount of excitation that you're averaging. Does that make sense? Here, when they're separated, and if you move it a little to the side, you would get a different input because every cone here is connected to a different ganglion cell. So when a lot of averaging takes place, very different inputs could give you the same output. And you know that because when we practiced those networks, you saw that you could get the same neural firing rate for different inputs. What does that mean? Well, if so many things are converge into one cell, you could have so many different patterns of visual input that give you the same result. But if everything is connected to only one cell, then any small changes can change the output. And that's what it means. When you have low convergence, you maintain a spatial resolution. As I said, rods and cones are distributed very differently. And again, you could see it here. Like if this is the fovea and the dark shading shows the distribution of cones, most of the cones are in the center. And the rods are in the periphery. So what that does, given that cones have almost one-to-one relationship to ganglion cells, it happens that, well, you get high resolution in the fovea. All right, they're wired differently. Now, but the rods are in the periphery and they're much more light sensitive, but they're not color sensitive. Now, what does that mean when I say they're not color sensitive? Based on what you learned last week. What does it mean? Okay. If they're not color sensitive, that if you lose your cones, you're not going to perceive color anymore. What does that mean? What does that tell you about rods? Yes. But what does it mean in terms of physical? What does it mean in terms of their sensitivity? What are they sensitive to? And what are they not sensitive to? How much light there is, intensity, yes, but does that define color? Okay. Yeah, so they're not sensitive to the wavelength of the light. They're only sensitive to the intensity of the light. So if a light comes from, right, if a reflection of light from those seeds activates the rods the same way as a reflection from this cap, right? It means that they're not differentially sensitive to the wavelength. They're sensitive to light, but they're not differentially sensitive to the wavelength of the visible light. As I said, again, this is the big picture you should all kind of internalize, right? If it has to be sensitive to that in order for perception to build on top of it. Now, rods mediate night vision because, well, they're much more light sensitive, and if you because they're much more light sensitive, they could respond to little light. And so when you're in a dark environment, they really help you. And experimentally, people have shown that you're more light sensitive in the periphery than you are in your central vision. So if you're looking at a screen and someone flashes a light in the periphery, you're more sensitive to it. Why? Because there are more rods in the periphery. So when you're looking at the place and you flash it, that light is going to be detected by some rods in your periphery. So you're more sensitive to it. We call it scotopic vision. Now there's photopic vision, which is the day vision, and it's mediated by cones. And that's when you want to look at objects when you're trying to perceive them. So just side by side, rods are in the mostly mediate peripheral vision. And because they average a lot of the stuff, they have poor spatial resolution. There's a lot more of them than cones there are. They're more light sensitive, so your night vision really relies on the sensitivity of rods. They function in the dim light for that reason, and they're achromatic, which means they're not sensitive to different wavelengths of light. They're only sensitive to the presence of light. And that's important, right? If they're not sensitive to that, then you're not going to have color vision. Again, there is a more higher level concept for you to think about is, as I said, color is not the property of that chair, right? Color is the differentiation of wavelength that you're able to detect. That's what color is. Cones are mostly in the central vision. As I said, there are not as many as there are rods. They have very good spatial resolution because they have lower convergence factor to the ganglion cells. They're not being averaged out. Every cone, whatever it detects, it passes on to the ganglion cells. They're less light sensitive. They function mostly in the daylight, and they mediate color vision. Okay? So let's think about, what is the process of transduction in the eye, right? What kind of energy is becoming what? So what is happening in the eye in terms of energy? What is it? Come on, guys. What kind of energy is being, yeah. Yeah, so it's light energy, and it becoming what? Neural activity, right? If the brain can only works with neural activity, with the spikes, with firing rates, that's the only thing the brain can actually work with. So the process of transduction is taking the physical energy in the world, light, electromagnetic energy, and converting that energy into neural activity. So how does that happen in the eye? Well, it happens in the rods and the cones. So there are these pigments that are somewhat unstable. These are unstable proteins. And for the rods, they're called rhodopsins. What happens when a photon of light hits them? Well, they change shape. Something like this. So proteins can change shape with energy. In this case, this protein changes a certain shape when a photon of light hits it. What does that do? Well, it opens up certain channels. When the shape of a protein changes, it opens up certain channels. And it actually hyperpolarizes the photoreceptor cell. So you would think that light activates a photoreceptor, but it's the opposite of that. Photoreceptors are generally active, like they have a base. Again, the reason we went through a lot of those practices for you to understand what baseline activity is, that every unit, every cell has a baseline activity. And sometimes the way to activate something down the line is to inhibit something. So if you inhibit the photoreceptor through light, if I told you that it makes the bipolar cells more active, what would that tell you about the connection between the photoreceptor and the bipolar cell? What does it tell you about? It's inhibitory. So you could say, well, these photoreceptors have an inhibitory connection to these middle layer cells, and there are a bunch of them, but let's say we just call them middle layer cells. If the connection between these receptors to these were inhibitory, what would inhibiting the photoreceptors do for the activity of these cells? Increase it. Because, again, I'm going to go with the metaphor again. In a normal way, the photoreceptors is putting a brake on the middle layer cells. It's like, inhibiting them. When you inhibit this one, it means that you're picking your foot off the brake. You're inhibiting because it was really, inhibiting those cells, and then you just pick your foot up by putting light on it, and then the cells could become more active. So you're inhibiting it, and it was inhibitory for another thing, and that's the negative one times negative one. You remember that from exam one, and that becomes a plus one. Yeah? Are you all with me on the yeah, so light inhibits the photoreceptors. That's what it does. It kind of, when you shine a light on a photopigment, it changes shape, it opens up a channel, it hyperpolarizes the cell. It hyperpolarizes it, it reduces its activity, and because this one was inhibiting something else, that one comes up. So it's like a seesaw in that sense. And that's the process of sensory transduction. That's how light becomes normal activity. And then when the light goes away, the protein goes back to its original form. So, and the process continues. And we have a lot of these rods, and we have a lot of these, photopigments that are on the surface of the rod, and that's how it works. Questions? Yeah.
Would you say it's sort of like how an action potential works?
Action potential in what sense? So this would be an action. So the photoreceptors have action potential, right? All these cells have action potential. So the photoreceptors are constantly active. They have a baseline activity. It means that they're fired with a certain rate. The middle layer cells are firing with a certain rate, and the ganglion cells also firing with a certain rate. And the light kind of changes that makeup of how the action potentials happen. So when you shine light, so if the light is coming down here and hitting, let's say, these photoreceptors, it inhibits them. It reduces their firing rate. That in turn increases the firing rate of these cells, and then it increases the firing rate of the ganglion cells. So they're all cells, they're all neurons, and they're firing. And you listen to them, like that's how they're behaving. They're just constantly firing. Sometimes they're above their baseline, sometimes they're below their baseline. But the thing is, how they're connected to one another, whether the connection is excitatory or inhibitory, changes how they interact with one another, right? And I hope that you all got that from the first, module of the class, that the communication between the cells. That was what we wanted you to take away from that module. How do cells communicate with one another? How they inhibit and excite one another? Okay, but that's an interesting, yes.
So for people who wear glasses,
Yeah.
So is that like a rod and cone problem?
No, that's more of a lens problem. So this lens, in order to bring something into focus, right? If I don't fall down, if I am at a certain distance from this text, right? The zoom goes out, like your camera, right? If you move your camera closer or farther from something, it goes in and out of zoom, which means out of focus. Why? Because when you have a lens and you played around with magnifying glass and whatnot, right? The lens bends the light. It bends the light in order for you, for the fovea to be the focus. Basically, that's what it does. But for some people, the lens gets harder as you age, and you cannot change the shape of the lens. So a lot of people need glasses because of that, and that's why you get surgery. You could change the shape of the lens. It brings certain distance into. So a lot of it has to do with that.
So it's like a problem of the lens manipulating the light.
Yes. Yeah. So it's a problem of focusing. Yeah. Yeah. So color blindness has to do with cones. Like some people genetically don't have certain cones. And if you don't have that, it creates. So, when you watch TV, you all have seen it up close TV, like older TVs, it was very obvious. How many pigments do you have? Three, right? You have three pigments. You need three in order to have color vision. You need three elementary colors. It could be blue, red, green, for example, right? It could be cyan, magenta, or yellow. It has to cover the spectrum. And with combining those three, you could get color vision. If it's only two of them, you get this. I don't know if anyone here has color blindness, but you get this. For some people, you get this spectrum of yellow to blue. Like it's like everything is in the spectrum of yellow to blue, for example. Um, and that's because you don't have one of the cones. And then with two, you cannot really get color vision. So that's the problem. Yeah. That's yeah. I mean, the structure of nothing is that complete until certain age, if that's what you mean. But what I could imagine that being true. Yeah. Yeah. No, I'm just saying, yeah. Like children's perception for ability. I think so. Sometimes those claims are um, over, let's say, overclaimed. What they say that people think that maybe a child only see blurry stuff or whatnot, but it's much better. The evidence shows that it's much better than what people think it is. But I think by the time you're like 2 or 3 years old, you probably have a really good like vision, like as good as an adult probably. It's just that you need expertise to dissociate the stuff, but your vision is probably as good as an adult. Um, yeah, so the eye, there is a lot about the eye that is interesting, right? It's and again, you mentioned like glasses or something, or there are a lot of like ailments of the eye that you have to, correct for. If you could only see farther stuff or you could only see closer stuff, all of those things have to do with how you're bending light. And we're not talking much about it in this class. We don't have time to get into the ailments of the eye and whatnot. That's a whole class for itself. Um, and all the ways where something could go wrong and yeah, but the idea is that if you played around with the lens or if you have some idea of what focus is, that's what it is, right? You have to focus. If I'm talking to someone in the front row, if I'm talking in the back row, I'm using a very different, straining on my lens in order to focus on their facial expression or something like that. And that's because when light comes, you have to bend it, and it's as it gets closer to you, you have to bend it more and more in order to because it's diverging, and you have to bend it to make it the same. All right. Now, another interesting thing about the eye is something called a foveal pit, and we're going to talk about that. Now, fovea is a special place in the eye, and that's what I meant. Like, as I said, most of our actions in a day is to bring something to our fovea, right? So if I'm moving my head and I'm moving my body and eye, in order for this specific letter like E to fall on my fovea, that's, because fovea, that again, playing around with lenses, there is, each lens has an optical axis, and that optical axis is a place when light does not get distorted. It goes through the lens with no distortion. So any lens has an optical axis where the light could travel, and that's where the fovea would be. So that's where the light travels without any bending. Um, that's the place that there are a lot of cones. But there's something very special about it, and you could see it here in every figure that I have. Uh, there is a little pit here. You see that? There's a little bending of the cells in the fovea. Why would that be? Someone take a guess. I have this slide there, but take a guess. Again, this again has to do with the organization of the eye. The fact that the receptors are at the bottom. Did you want to say something? Or?
Oh, I was just gonna say that because there's more cones at the phobia and there's less
Mm-hmm. Good guess, but no, that's not the reason. The reason is, again, has to do with this organization of the eye, where the cells are. So as you could see, light has to travel through the ganglion cell, through the middle layer cells to reach the receptors. You can't do anything about that in the periphery. But what you could do in the fovea, you could basically part them, right? This is like Moses and the Red Sea. You part the ganglion cells. You just create a dip where the light could travel without any hitting any cells and reach the photoreceptors. Okay, so that's what a foveal dip is. It kind of allows for the light to travel without hitting anything and go right to the photoreceptors. And this is again a side effect of having this organization where the light has to travel through the cells in order to reach the photoreceptors. But fovea, as I said, is a special place for many reasons. It just has, and I mentioned a few of them just now. One, well, there's a foveal pit in order to part the cells, in order for the light to reach there without hitting any cell bodies, because, well, that would distort the light. It is where a lot of cones are, where it gives you color vision. This is where the convergence is lower, so you could get the highest resolution. So you're not averaging a lot of information to lose resolution. This is where the optical axis of the lens falls. So you're not bending the light in order to reach the fovea, right? So for all those reasons, we get to have this really high resolution perception of the world at our fovea, right? That's why we have to look at things in order to recognize them. And if you want to do something that requires really fine details, like reading would not have been possible with these tiny letters that we use, in on our phone, for example, if it wasn't how high resolution for all those reasons. Yeah.
Is there like an evolutionary reason why it's organized with like ganglion cells, secondary cells, and photoreceptors, and not just the opposite? So you don't have to pass through all these secondary cells. I don't understand anatomically why.
Well, I think the best way to explain it is that sometimes, like from an evolutionary perspective, right? Sometimes you find local optimals, right? Meaning sometimes you stumble upon a solution and you just stick with it, right? So if if an animal evolved this type of organization and it worked and it was good enough, And over the years, other stuff happened, like the parting of the this and that and that and that. Then it kind of worked for that animal. It didn't require a whole other. See what I mean? Sometimes this is something how people interpret this, that you come upon a good enough solution and then you stick with it.
So is it possible that animals who evolved eyes in different phylogenetic
Yeah,
groups
possible.
and
Yeah,
also had the reverse organization?
it's possible. Yeah, but I guess our line of evolution all have, like primates and all those other, have this sorts of organization. Um, but yeah, I think it's just a matter of a solution that is good enough could persist. Um, because again, it's a matter of optimization of a system and everything. Like we all do that, right? A good enough solution sometimes works and then you stick with it. And I think evolution does a lot of that. Like a good enough solution, just you stick with it and then it evolves, because it's not an intelligent design supposedly, right? It's just through these sorts of uh, evolutionary steps that it comes about. All right. Yeah, for all those reasons, phobia is a special place. It has the highest visual acuity. And this is another way of saying high resolution. When I say acuity, it's really resolution, that you have a really high resolution and you could process information that are really small and like close to each other and everything, because you could see everything. And when I say resolution, I'm really talking about when people talk about resolution in an image, right? If an image is low resolution, what do you lose in that? What kind of information do you lose when an image is low resolution? Details. So imagine those details aren't available in the periphery, because all the visual input in the periphery is like that. And as you get farther and farther from the fovea, they become worse. Like they become lower and lower resolution. So we'll talk about it more in a few weeks. The world seems kind of, high resolution in the periphery and stuff, but it's not the reality, right? If it's the reality of peripheral vision is really blurry. Um, so visual acuity is another name for high resolution details. Um, and in order to have the clearest view of an object, we want an image. We really want the light from that object to reflect on our fovea. All right. Let's take a 10-minute break and then we'll come back.
If you're here and you didn't sign the little sign-in sheet, then tell me that you're here.
Okay, so the organization of the retina, that's the first place in the visual pathway, basically, right? That's the first step. It's a sensation. You wanna convert the visual input, the lights in the world, into neural activity. Now, what happens to this light? And I said, this is the property of light, and when it hits a lens, what it does really, and again, if you played around with a lens or a peephole or something, light gets flipped. So when you're looking at a pen, if I'm looking at an object, the light from the top kind of gets flipped and then goes to the bottom part of the retina, and then from the bottom. So this flipping in space does happen for every visual input, basically. And again, this is because there is a lens, and lenses do that. And you could imagine how with distance this changes, right? If something's farther, it would land on a smaller part of your retina. And if it's really close to you, it's covering a lot of your retina. Another thing that lens does is it's also flipping things horizontally. So So if this is a person looking at the visual world, there is the right visual field and there is the left visual field. Uh, and you have basically input coming from the right visual field, input coming from the left visual field. You could imagine that this sort of flipping would happen in both eyes. That the input from the right visual world, and I'm showing that with green in this schematic, would fall on the temporal part of the left eye. So this is input from this part of the world. It's if it's only hitting my left eye, it would fall on the outside of the eye, which is the temporal part. And then the inside, we call it the nasal part. Just think of it, this is close to your nose, so we call it the nasal. The same visual input, when it hits the right eye, it falls on the nasal part of the right eye. So this flipping that I was just describing happens horizontally for both eyes. The visual input comes, it gets flipped through the lens on this side, and then flipped through the lens on this side to the temporal side. Now, what the brain does is that it actually gathers information through the optic nerves, and these are the nerves leaving the eye, and these are all the axons from the ganglion cells that you saw. All of them are leaving the eye through the blind spot, through the optic nerve. And when they're coming from the two eyes, those remain separate. They remain separate, and then all the visual input from the right visual field, which is in green, comes and goes, gets gathered, and this is a place called optic chiasm, where all the visual input in green gets gathered, goes to something called LGN, lateral geniculate nucleus, and together they go to the left hemisphere of the brain. So when we say the left hemisphere of the brain is processing the visual input from the right visual world, this is what we're talking about. Because all the nerve fibers, all the axons that are actually carrying information from the eye that are processing, that are responding to the right visual world are being gathered and then going to the left hemisphere. And the same works for the right hemisphere in the brain. What it does, all these information that are coming from the left visual field is coming, so these are colored red, and the information coming from this part of the world falls on the temporal part of the right eye and on the nasal part of the left eye, all coming from the left hemisphere. They also go down and they get gathered in the LGN, and then they go down to the primary visual cortex. This is one of the pathways. And this pathway is called genicular striate visual pathway. It connects the retina to the lateral geniculate nucleus, and that's the step along the way before going from the LGN to the primary visual cortex. And I said this is the much bigger of the two pathways, and most of the visual processing basically happens here. The lateral geniculate nucleus is it's a very small area, and it's bilateral. When we say it's bilateral, it means that it exists in both sides of the brain. You have one in the left brain hemisphere and one in the right brain hemisphere. That's what bilateral means. And that's the only place that the neurons actually stop. So you have all these axons leaving the eye, they get to that point. From that point, they go to the visual cortex directly. And as you remember, the visual cortex is in the back. Now, so this visualization here is basically slicing the brain this way. It's a slicing it this way at the eye level. So if I slice it this way on the eye, it just travels to the back of the brain where the visual brain is. As I said, what you need to do in terms of anatomy is really, and the optic chiasm is basically where there is this crossover, where the visual input from the either hemisphere are crossing over in order to go to the other side. In terms of anatomy, I really don't want you to memorize a lot of stuff, but the projection of the activity from the LGN to the primary visual cortex is called optic radiation. Superior colliculus is part of the other pathway that we're not going to talk about right now. We'll talk about it later. But the pathway that we're interested in right now, which is the pathway where a lot of pattern recognition, all of those stuff happens, is the pathway that you go from the eye to the LGN, and from LGN, you go to the primary visual cortex. Good. So we already saw the response that the ganglion cells could have. Like a lot of the center surround responses that I talked about are ganglion cell responses. And they basically are either, as you saw, they're on center off surround, which means that the visual input in the center can really activate them. So if you have, let's say, so when you look at this, is this, and imagine the gray area shows the light. Is this a on center off surround or off center on surround? On center off surround. Because when there is light in the center and it fills up the center area, the activity and the way we show the activity of cell visually is usually like this. We're showing the action potentials. Each of them is one action potential, one firing of the cell. So the firing rate of the cell is much higher here than it is here. So because the excitatory is filled in, as soon as the light gets big enough to go into the inhibitory area, then the activity goes down. And in general, when the entire receptor field is filled in, your activity is lower than, well, you don't see it here, but I'm not going to say that. So, but think about it that this cell is excitatory center, inhibitory surround. It could have been the opposite, but we also have another name for it, which is, on center, off surround. So in a way, we're saying that, well, the ganglion cells are taking this information and taking it to LGN, and LGN has a very, very similar response than the ganglion cell. So there's not much of a change in the receptive field of the cells. But these are new cells, right? So there were some cells, these cells are connected to some other cells, and these cells have similar receptive field. Okay. As we said, we could have on-center off-surround or off-center on-surround, but there's also other class of cells that we didn't spend that much time on. So again, this is the firing rate, and this is the stimulus. So if the stimulus is coming on here, this would be, well, excitatory center, inhibitory surround, because when the stimulus comes on, the firing rate goes up. Here, when the stimulus comes up, the firing rate goes down. So because the stimulus is here. What happens here? How would you describe this response?
Firing rate increases based on a change from light to no light.
Exactly. So it's a change detection behavior. So when there is no light, the light comes on, the cell becomes active, but when the light stays on, the cells kind of starts going down again. When the light goes off, here, the cell becomes active again. So in a way, it's responding to the change in the light. So these cells are called on-off, something like that. So you could have on-center, off-center, or on-off, a cell that really responds to the change in light. There are all sorts of cells. Again, we talk about some of them, but there are a lot of other different kind of cells in the visual system. Okay. So something that is very interesting about the visual system is it's the idea of the spatial organization of it. Now, we're going to talk about this a lot. What's something that's interesting, let's say people found out if you take part of the visual cortex and you maybe zap it or cause lesion to it by some electricity or something. You could do that. You could do that to part of the visual cortex. You could create a hole in people's perception, right? Or a monkey's perception. They become blind to certain areas of the world because they're not processing information from that. If you now make the lesion bigger, what happens is that that area of blindness becomes bigger. And this is very important. It's important because it tells you something. What does it tell you? It tells you that these cells in the cortex that are close to each other are processing areas that are close to each other in the visual world. Does that make sense to everyone? It's an important property of the visual system, where the tissues that are close to each other are processing the same visual areas in the world. So if I basically take a recording from some area and I find the receptive field, and the receptive field is somewhere in the world, if I move the electrode a little bit, this receptive field is not going to jump to another place. Does that make sense? The receptive field is going to be next to it because these cells are next to each other in the cortex. In the tissue of the cortex, the cells are close to each other. So the receptive field, the area of the world that they respond to, are close to each other. And that's called retinotopic mapping. You all clear with what retinotopic mapping is? That the spatial information is preserved in a form of topographic maps. In this case, is a retinotopic map. We call it retinotopic. Why? Because, well, when I say two things are close to each other in the visual field, it is guaranteed that they're going to be close to each other in my retina. It's guaranteed, right? If I'm looking at this pen, if I'm looking at this cap, this area and this area are going to be represented by close by cells in my retina, right? Because the light from this is only slightly displaced from light from here. So the areas that are close to each other on the object fall on the same area of the retina. And that's why it's a retinotopic, because it preserves the organization of the retina. When two things are close to each other in the retina, they're also close to each other in the visual cortex. Now, this didn't have to be the case. You could have, so for example, when we talked about here, when you have these inputs from the two eyes being gathered in the LGN and through this optic radiation going to the visual cortex, it could have been jumbled, but it's not. Things that are close to each other, they're going to stay close to each other in that radiation. So two ganglion cells that are close to each other in the retina going to have projections that are close to each other in the visual cortex. I'm saying it in very different ways. I want you to understand it's an important concept. And you could, as I said, you could show that with these cuts by lesioning. If you lesion a certain area, and if you lesion an area next to it, the blindness is going to grow. They're not going to suddenly become blind in a very different place because the organization is retinotopic. Yeah. Say that louder. I didn't hear. Change in vision? So are we going back to, you mean in this case? So this is on the level of the eye. So in the eye, I was talking at the time, in the eye, what happens? If a light, so there are proteins, so rods and cones, they're photoreceptors because they have photopigments on the surface. It's a cell, like any cell, it has a surface. And there are photopigments on the surface. When the light hits those proteins on the surface, it changes their shape. The protein changes shape. When protein changes shape, it hyperpolarizes the cell. So in a way, an hyperpolarization of a cell means it reduces its activity. So that was the moment, that's the moment where light becomes neural activity. Is that the question? I'm not sure. Okay. But that is in the eye. So when in the eye, the light becomes neural activity, the neural activity now travels the brain. That's what I'm saying. So if in the eye that becomes neural activity, sorry. So in the eye, when the light hits the eye, right, like a big pizza pipe, you know the sound. Uh, when the light hits the eye, it becomes neural activity in the eye. That's the first thing it does. It hits the photoreceptors and becomes neural activity. And that neural activity, right? What we talked about it. We did some practice on this. When a photoreceptor has a neural activity, the activity changes the activity of cells that are connected to it. That activity changes the activity of cells that are connected to it, right? It's a cascade of layers that are connected to one another. So the activity in one layer gives you an activity on the next layer, gives you an activity on the next layer. And that's what we're trying to figure out for the vision. What happens to the activity that is generated in the eye based on the light? That activity travels outside the eye through these axons from the ganglion cells and impacts the activity of cells in the LGN. The activity of cells in the LGN impacts the activity of cells in the primary visual cortex. So at that point, when we leave the eye, we don't worry about the physics of the world anymore. We don't worry because we already got our neural activity. The light is converted already. Does that make sense to everyone? The light is converted to neural activity. So now the brain can work with it, can try to compute different things with it. So as I said, the eye changes because there is a lens. Again, this is all function of the lens. Because there is a lens, the optic of the world changes in the eye. So if you're looking at this, you're looking at this figure up here with the triangle and the rectangle or a square. In the right eye, what happens? Well, you have the nasal part. The nasal part is in the middle. You have the right eye and you have the left eye. This rectangle or a square falls on the lower half, right, on the right lower half of the right eye. Because, well, as I said, it flips the visual world horizontally and vertically. That's what the lens does. It flips the world horizontally and vertically. In the left eye, also it's flipping it horizontally and vertically. It's one visual input, but I'm just showing two of them because the eyes are looking at it at a slightly different angle. So this organization is maintained throughout the brain. The fact that the eye changes the organization of the world, that organization stays in the eye, meaning that this triangle will be processed with your left hemisphere. This square goes to the right hemisphere. Does that make sense? So that's retina topic organization because whatever happened in the retina is maintained throughout the brain. So objects appearing in the left visual field are represented in the right hemisphere of the brain and vice versa. You all understand how that happens now, right? Yeah.
So taking it back to the visual neglect that we learned about, is that damage to the LGN in the right hemisphere?
No, so damage to when the damage is closer to the sensory input, you're aware of the damage. Meaning that if I take and damage some part of your retina or part of the LGN, then you see a hole. See what I mean? So the damage is more perceptually, you're aware of the damage. If the damage is in, let's say, parietal area, you may have neglect, but you may not even know there is something out there, that you're missing something.
But that area, I guess, and you're looking in the brain, that is kind of over the LGN though, right?
So that's way down the line. So no, so we're very close to the eye still, right? So this is like where we are right now is like 2 steps away from the eye. So the eye takes the information, passes on to LGN, it goes to the primary visual cortex. These are the lower level visual pathways. There are then higher level visual pathways that that do different things, and the parietal area is part of the higher level visual pathway that are entirely in the cortex. But as I said, this is this sort of damage to the earlier visual areas, you're usually aware of them. Like meaning that if someone, damaged the primary visual cortex, you'll see a hole, like you see a little dark hole in this area of the world. It doesn't cause neglect. It causes blindness to part of the world. All right. Now, again, we are very close to the eye itself, and I want you to remember that we're very close to the eye. Like we're still in the level of very simple processing. We haven't even reached, orientation detection that we talked about last time. We haven't even reached there. We're still in receptive fields that are just circular, center surround. Now, the input from the LGN projects to the back of the brain. So the LGN is in the center of the brain, like the eye, the input from the eye goes to the LGN. The LGN then just projects that into the back of the brain. There are a lot of nerve cells leaving the LGN, going to the primary visual cortex. And this is what's called optic radiation. It's a bilateral structure, meaning that you have one primary visual cortex in the right hemisphere of the brain, you have another one in the left hemisphere of the brain. And it's very large compared to LGN. It's one of the largest areas that we have in the brain in terms of processing. And this is where a lot of those low-level visual processing takes place. Like the video that we were watching last time, a lot of it, those simple cells, the oriented bars that they're responding to, these are all in the primary visual cortex. But let's look at a few figures. I want you to get a mental model of retinopathy. So if I have a visual input like this, and this is the surface of the cortex. Should I show a different one? So let's say you're looking at this picture, and basically, if you're looking at this center location where my mouse is, it means that which hemisphere of the brain would be looking at this area? The right hemisphere, because this is from the left visual field. And this is showing the right hemisphere. So if you look at this figure, this is the right visual field. So it would project into the left hemisphere. But the projection is not one-to-one. And we'll talk about this next week. But I just want you to have an idea. So also, not only this is projected to the right hemisphere, it is flipped in the sense that it's the eye goes at the bottom part, the nose is, you see what I mean? By flipping. And it's distorted in a lot of different ways. So the projection, again, this figure is just a schematic, trying to show you how visual input theoretically, like not theoretically, but how would they respond? They don't exactly respond to this, right? Because they're going through processing. It's another figure. This is a better one. So this is the right hemisphere of the brain. And back here, so imagine there would be two of them side by side, the right hemisphere and the left hemisphere. And side by side, um, and this is the right hemisphere. So it is processing visual input from the left side of the brain. So this figure should give you a good. So if you're looking at this center point, the blue area is in the upper visual field, so it would project to the lower part of the V1, and this colorful area is in the lower visual field, projecting to the higher part of the V1. But as you can see, it's flipped in a sense that this is part of the left visual field projecting into the right hemisphere, but also flipping that way. I'm just trying to give you some visualization of what it means when I say the visual input projects this way. Again, there's a lot of processing happening, right? These coloring don't mean much other than the fact that you're saying if you look at a cell in this area, its receptive field is likely to be in this area of the visual field. That's what that means. Hopefully that helps you. I'm not sure. To get a mental model. I'm going to talk a little about the organization of the cortex. I know that Ellen gave you an overview of anatomy and stuff like that, but I'm going to just talk a few minutes about it so all of these things would make a little more sense, hopefully. So the cortex really is not, so if you take out the cortex and you spread it out, it's really the size of a large dinner napkin. And its thickness is about a few millimeters. What happens is that when you try to bunch up that dinner napkin in the brain, you get something like this. So if you actually take a brain, and you should all kind of be understanding that, well, this is the front, this is the back, this is the brain stem. So this would be frontal lobe, parietal lobe, and temporal lobe, and like the occipital lobe in the back. If you slice it the way I'm slicing it here, you would see something like this. You would see that dinner napkin kind of on the surface, and everything else is the connections. So it's basically the surface of that. And all the recording that people do from the cortex is really only a few millimeters of the surface of the cortex. It doesn't go deeper than that. Under a microscope, it would look something like this, that you have this surface stuff. And the thickness of it is really small. So this is a slice this way. If you slice it that way, so horizontally you slice it, you would get something like this. It's the thickness of it is small. Everything in the middle is just connections. And on the surface, you have a small. So when we talk about a primary visual area having a retinotopic map, it means that it's projecting on the surface, not in depth, but on the surface. So the visual input is projecting there, and you kind of could see, because it's a flat surface, you kind of could understand maybe what does it mean to maintain the organization of the retina. Does that make more sense? It's like a flat surface that is bunched up. So the whole thing is a flat surface that is bunched up, but the V1 is like a smaller flat surface that is bunched up. So, if you open it up, you could kind of imagine. What's happening here is that you kind of blow up. Imagine you're putting air in one of them. So the napkin becomes kind of blown up, so you could see all the areas, because in this case, as you could see, there are a lot of nooks and sulci and gyri, and you cannot see everything. If you put air in it and kind of open up this thing, so you could look at everything, kind of open it up, you would get something like this. Kind of blow it up. So all the areas that are protruded, or as I said, gyroid and the stuff that are inside, like the sulcus, then they would just blow up. Okay. So you see, this is just a different visualization of this thing. It's just that it's spread out so you could see all the surfaces of the cortex. Does that make sense? Does that help? Just imagining what we mean when we talk about the cortex. All right. Any questions about this?
Question.
Yeah.
Can you say something like a retina topic organization? What are other, like, isn't that kind of everything if we're seeing everything?
Not all areas are retinotopic.
Okay.
So some areas are retinotopic, some areas are not, meaning that you could respond, like when you get farther from the retina, as we talk about it, it's not really that, okay, that the receptive field is only from a specific area of the image and that anything next to it or next to it, right? That organization falls apart.
Okay.
But it's like an area to area.
Okay.
But also in the brain, it didn't have to be like this. It could have been more jumbled. Okay, so the primary visual area is really the last stop in this genicular striate pathway. There are higher level visual pathways, and those are the ones that we're going to talk about next week. But in this lower level, and when we talk about level, we're really talking about how close you are to your sensory modality. If you're really close to the sensory modality, like the eye, so these are lower level pathways. That's really leveling means. And it's a bilateral structure, as I said. It's very large. It's on both sides. So you could see that on the surface here, this would be all. So I'm hoping you're getting a good intuition of this. But so imagine this area would all be V1. So basically this area would all be V1. So it's on the surface of the cortex. It's only a few millimeters thick. And it's like, let's say, maybe a couple of centimeters by a couple of centimeters, and it has a little thickness. That's what we're talking about when we talk about V1. But because it's bunched up, it looks kind of confusing. But in reality, it's just the surface. It's like, imagine, as I said, imagine I don't know, the size of an ID card with a little more thickness. And that's what V1 is. And all the brain areas, all the cortex that we're talking about, they're really just little surfaces. They're just little pieces on this surface. That's what they are. And because this is a surface, it can maintain that retinotopic organization. You could really say, okay, these areas are close to each other in the retina. I'm going to have them close to each other if we want to. Something like that. But as I said, it's a surface, but it's also had a small like thickness, a few millimeters. So those few millimeters could really be layered. Even, imagine you have a really thin dinner napkin and you have one on top of that, one on top of that. So you have like 6 or 7 of them on top of each other. And each of them maybe are, half a millimeter. So the whole thing is like 3-4 millimeters. That's what cortex is. And the LGN really projects only to layer 4C. So you have 1, 2, 3, 4 has 3 of them, and then 5 and 6. And really it only projects to layer 4C. So not only that the cortex is layered, but also all these LGN, the projection that comes, it only comes to a certain layer. So, and the cells in this layer, now again, so think of it as layers. So you have a layer in the retina, our photoreceptors, you have a layer that are middle layer cells, and then you have a layer that are ganglion cells, and another layer is in the LGN, and then there's another layer in the primary visual cortex layer, you know. So basically, a signal is being processed at each stage, a little more, a little more, a little more. And the cells in layer 4C have the same center surround receptive field as the LGN and the retina. They don't really do extra processing. So cells with more complex receptive fields are found in the V1, but in the surrounding area. So it's not in the layer 4C, but we have what we call simple cells, complex cells, and ended stop cells. You saw examples of the simple cells already, right? This is an example that I use. This is what a simple cell is, and it is in V1. We saw how it responds from that video, that it responds to a specific, and these are repeats of what we had. It responds to a specific orientation of light. For example, this one, it was on the exam. It responds to a certain orientation of light, to vertical orientation of light. It doesn't respond as much if the light is horizontal, and as I talked about, again, this was on the exam too. It is constructed by having these simpler receptive field in the area 4C of the cortex. Like the visual cortex has responses like these, and it can combine them in order to create more complex. Now, these functions as feature detectors. We talked about this. If you can detect certain orientation of light, it means that you could detect edges. And when you could detect edges, you could put them together and then detect something else. So again, keep in mind, there's hierarchy, and we're trying to build the hierarchy from the retina, from the photoreceptors. What is the hierarchy of processing? And we started very early in the retina, in the photoreceptor, say, okay, you build a little hierarchy, a little on top of that, a little on top of that. And as you go layer by layer, you could build more and more complex visual patterns that you could respond to. And we'll talk about that more next week.